you a question about about the 100 different proteins. I can't really do that. So what I'll do is I'll pull topics that we've discussed that they talked about during their talks. All right. Anybody coming to the department Christmas party?
Is it extra credit?
What? Oh. Is it? You got the?
It's just
Is
for
this
fun.
the white elephant?
Yes.
Yeah.
Christmas.
Are you going?
Yes, I made buffalo chicken dip,
Oh my
which is
God.
it's got cream cheese,
Wow.
cheddar cheese, ranch, and I use Frank's buffalo sauce. You mix it all up and you dip it in chips. It's delicious if you like things spicy. Yeah, it's so easy to make, and all you do is mix it, and it's so delicious.
That's the best kind.
It has chicken.
I'm going, but I'm leaving early though.
What's that?
I'm going, but I'm leaving early
You're leaving early. Why?
to do something
What
else.
is stoic make?
Yeah.
I don't know.
Like boiled beets or something?
That's so rude.
Okay, go for it.
She tries to be so nice, y'all.
Yes, it's very nice.
Apparently it's Ukrainian. Boiled beets?
It's just the European, Eastern European kind
She
of,
can't beat that.
you know, I don't know, sternness, right?
Yes,
Yeah.
she's
She's
so
very
nice.
nice. She's very
She's so nice, and like, I feel like she does not.
Yeah.
Sometimes that some words,
Yeah.
just like by the very nature of the word, can be offensive.
Yeah. So let me see here. So,
Like that time she told me that.
here, for the first chapter that we
Yeah,
covered, I
that's
think
your
that was
specific copy back
21,
from our old
okay?
class.
What do you need to focus on? That part was on the genome, okay? So we talked about the organization of two different types of genomes. We talked about prokaryotes, okay, and the nucleoid, okay? And then on the other hand, we talked about eukaryotes and the chromatin, right? And how that is organizing the chromosomes. So, what do you want to study for those? Can you explain those two different ways of packaging up a genome? So for the chromatin, right, we have the histones, we wrap DNA around it, and we make the nucleosomes. This is related to what Chad and Brandon talked about in their presentation, okay? They talked about that. So do you know the organization of eukaryotes into nucleosomes and to chromatin? That really isn't a whole lot. I mean, just knowing like the histones, okay, they wrap around, so those types of things. So, and that eukaryotes have introns and exons. Do you want me to go through the slides on these, or is this okay me just telling you?
It's okay,
This is
tell us.
fine, okay. The next one is DNA replication. What do you need to know for DNA replication? All the intricate details. No, the machinery that's required, okay, to do DNA replication. This is probably something you've done in many different classes, right? We have the replication fork, we have DNA polymerase. What does DNA polymerase require to actually polymerize and copy DNA? Who remembers? What's nucleotide? That's good, yep.
A primer.
A primer, okay. And how is that different from RNA polymerase? What does RNA polymerase require a primer?
No.
No, it needs
No.
a promoter region.
Yes, needs to have an exposed promoter region, right, to bind to. So I want you to understand just that machinery that's required to do polymerization. Remembering that eukaryotes are more complex, we're just gonna focus on, you know, basically the prokaryote system. The eukaryotic being having more components, right, much more complex. So just focus on that. Can you explain that and maybe draw out a schematic showing that? So that would be the replication. The next chapter was DNA repair, okay? And we went into a lot, there were all kinds of DNA repairs, right? Thymidine dimers, you can have the reverse process. I just want to boil it down to three. Nucleotide repair. Now, nucleotide repair, remember, is where you cut out a section of DNA and then fill it in afterwards, right? Comes along, it sees, say, a thymidine dimer, goes downstream, clips a spot, goes upstream, clips a spot, removes that larger piece of DNA, then uses the template strand and fills it in. Okay. The second type, base excision repair, cuts out an individual base. Usually that's from removing the base, leaving the sugar behind, then it clips out that sugar, backfills basically. Okay. The last one is, we talked about recombination, and recombination is used not only in repair but in other processes. But I just want you to focus on homologous recombination. Okay, that process. And let me pull up. We're in proteomics. I don't remember where I am sometimes. Okay. So there were different ones. We talked about heterologous, homologous, and then there were a lot of recombinations in the chapter after that. Okay, because it's actually used for increasing diversity, genetic diversity and things of that nature. So just the homologous one. So that's it from that chapter. Okay. Then we got to transcription. I already kind of gave it away. I want you to know how RNA polymerase works. Okay, what's it require? You know, things like it can slide, right? It has proofreading capabilities, and unlike DNA polymerase that continues, it can slide, right, back and to kind of proofread what it's already done to make sure that it has good fidelity. Oh yeah, you can't see a thing, can you? There we go. So our focus is on this one, nucleotide excision repair, base excision repair, which we showed here, and lastly, this, homologous recombination. Okay. Don't worry about this one over here, the non-homologous. That was it from that chapter. Okay. We talked about that genetic diversity. Okay, so then transcription was the next chapter. I just want you to understand how a transcript is made. I would like you to also know that transcription and translation are coupled in bacteria, but not eukaryotes. So I want you to always be aware of those differences and be able to answer questions about them. Okay, so that's transcription. Just focus on understanding that. There wasn't anything else that I really saw. In translation, transcription, we talked about bubbles and things of that nature. Like what we talked about, there has to be, you know, the promoter region has to be available. So focus on just, because it's the simpler one, the prokaryotic version of this. Prokaryote has to have an exposed promoter region that allows it to come in and start doing the transcription. So how does polymerase, RNA polymerase work, and what kind of allows it to start? All right, translation. Okay, I want you to know each stage of translation and the components involved. Okay, so there's the initiation phase, right? And it involves initiation factors. Okay, then there's the elongation that involves elongation factors. And then there's termination, which involves release factors. I think I call them TFs, termination factors. They're actually release factors. And if you look at it, there isn't a whole lot really there. There aren't a whole lot of factors to remember. You don't have to know, for instance, the recycling pathway for the elongation factors, you know, off of there. The multiplication factor TU, for instance, you don't know how, it has to know how it recycles. But you want to know that. So those different stages of how it's initiated, elongated, and terminated. And then gene regulation. I have no idea what I just wrote there. Let me pull that open. So information decoding, we know how that works. I want you to know the two kinds of control. And just use the lac operon, that's our model system. And how do we understand other systems? From a model system. So I want you to be able to explain an example of negative control of a gene, as shown by the lac operon. That's basically where the protein binds to it, right? That regulatory factor, that repressor protein. And then when you get the signal, it falls off, and that turns it off, or turns on the process. It's negative because the protein that regulates it doesn't bind to it when it gets the signal. It actually falls off when it gets the signal. So in the controlling factor. And then the other one would be the CRP, so the cyclic AMP receptor protein pathway. Where's that at? I just probably
That's
passed
the positive one.
as the positive one.
Mm-hmm.
So you can understand both of these. And so is it one or the other? It's actually can be a combination of these. Okay? So if you can understand these positive and negative controls, you can understand it for other systems. So this would be your positive control here. Okay. And of course, there are other controls that we talked about associated with things like ribose switches. That was kind of like this trip, that the RNA, right, can switch around by binding to certain things or having the presence of certain things. Okay?
Is this termination?
No.
Ribose switches?
So that can control whether it does terminate prematurely, right? It can. So, were you here that day?
Sure.
No, you
Yeah.
weren't. You're always asking me questions from the days you didn't come. I can tell you why you don't know. Okay, so this is the idea, right, that if it can bind to this region, it will influence the structure of it so that it can continue on or terminate early, prematurely. I would rather you know, what I want you to be aware of are what are ribose switches. Okay, that these are RNA molecules that bind to other molecules. Okay, you may remember me using a term aptamer. So the other molecule that it binds to is an aptamer. And what that can do is it can act as a molecular switch to turn things off or on. You don't have to know the specific pathway, but I really want you to know that there are dynamic control mechanisms in the RNA themselves. Epigenetics, be able to explain what epigenetics is. So no change in sequence, it's due to the I don't know, I want to say translational modification. It's
Post-trauma mark.
yeah, it's modification to the DNA, okay, that influences whether it can be expressed or not. And it's genetically passed on. Change in gene expression without a change in sequence. Okay, ribose switches we talked about. Okay, and then as a control mechanism, we know there is RNA editing. If you look at the bonus looked at three things that controls the expression of genes. And you can look at that, as an example of these different types.
What was the answer to that bonus? Because I don't think anybody ever got that right.
Did you look at did I post the answer key?
Yes.
Is there an answer? Is it on the answer key? There's your sign. Or there's your answer. Have you ever watched Doug Inville? It's like, He's a comedian? Okay, go watch him on YouTube sometime.
Did you
He's
post
like-
that on Canvas as well?
No, I didn't. But are you on Canvas?
Yeah, I got it. I wanna be.
Okay, there we go. I'm just making sure, because I remember we had this discussion before. All right, so that's what you need to study for those chapters. That should narrow it down so you're not frantically looking around. Now, for the topics that are really focused on proteomics, you have to know mass spec, okay? Not how to decipher it, but I want you to know the bottom-up approach for analyzing proteins using mass spec, MSMS. This was in pretty much most of your papers, okay? So do that. Mass spec, know how to explain how that works. Okay. So proteomics, finally. Okay, that must be it. Oh no, that was the ending one. Okay. Let's see here. Methods, that's what it was. Methodology. So in the methods, I want you to also know, since it was in Biochem 1, how to do 2D isoelectric focusing. I'm sorry. So those are the two main methods that we saw in all the papers that I went over and over and over again. 2D, right, isoelectric focusing. Okay, which we've seen before. And MSMS. Okay, we're doing bottom-up MSMS. Okay. And so here we have an SDS page. We could do 2D, right? Isoelectric focusing to look at a whole cell. That will help spread out the proteins more. But how we do MSMS, right? You can do this on either type. I focus on ESI, electrospray ionization. But basically you send through and do mass spec on peptides that have been cleaved up. Then you select one of those peaks, right? You select one of those peptides that you had seen in the mass spec, and then you send it back through into a collision cell, break it apart and start chopping it up in the mass spec, right, into smaller fragments, and you can look at the different peaks, and it will actually, from that, you can deduce what the sequence of it is. Then if you get the sequence, right, we can use bioinformatics backwards. So this was an example of using the quadrupole where we can select let everything fly through, we get a mass, okay, we select for one of these, send it through a collision cell where we bombard it, bust it up, and now we get fragments of that, and we can deconvolute what those fragments correspond to as a mass. This is another one. Okay, and then the bioinformatics. So this slide really tells you the flow of things. Okay, so in the process of doing bottom up, okay, you start with your sample, okay, then you usually digest it. Who remembers why we digest up the proteins? I talked about this last. Get in the gas phase better, gives us better signal, right? Because large proteins suck at flying into the gas phase, at least by ESI. By chopping them up, we get them into fragments that fly better, we get a better signal, okay? And that's when Dr. Chowdhury came, he was talking about that. We use the bottom up because we get better signal to noise ratio. Okay, so we usually have to digest it. We can enrich it if we want to get rid of background, right? As you're searching through all this stuff, it's like, oh my gosh, it's a forest of all these different proteins. Well, we want to focus on a specific problem within the cell. We want to look at maybe phosphorylation versus non-phosphorylation, okay? We can do some sort of enrichment for that so that we select out for what we're actually trying to look for. And of course, we could go through that flow that we already talked about. So,
Can you tell if one amino acid's phosphorylated once it's broken down, or if it only tells us that fragment has that change in
you can decipher it. So once you get the mass or figure out the sequence of the peptide, there might only be one amino acid on there that could be phosphorylated. So serines, tyrosines, threonines. Well, if there's only one serine on there, you could decipher, well, that's phosphorylated. Or you could even, if there are multiple serines, you could actually probably figure that out by the degradation. So going back to here. Okay, we cut off, we're breaking it apart, and we're actually getting the sequence. Well, you could figure out which fragment contains the phosphorylation and figure out which amino acid that has it too. So yeah, it's a powerful method. That's why I think it's really important for y'all to understand how to use MSMS. If you take anything home from this class, it would be how to use mass spectrometry to look at proteins globally. That's the point of proteomics. We can't just understand the problems that we're trying to decipher in the cell without getting a more global look at things. Studying one protein at a time gets us part of the way there. We need to understand not only what the proteins do, but their levels, right? And that's what proteomics is about. Well, we've done a real good job as biochemists of extracting proteins, seeing what they do, running kinetics. Okay, let's mix these two proteins together. Oh, this is what they do in unison. But if we want to understand the complexity of the cell and things that are happening, we have to not only know what those proteins do, but their levels, okay? Because that's going to influence the cell, the organism, and whatnot. Does that make sense? That's what we were trying to get out of that. So MSMS, 2D isoelectric focusing. We talked about that over and over again, how you can quantitate things. Okay, what would I like you to know? I'd like you to know SILAC, because I know for sure Chad's did that. Most of yours did this SILAC, right? Heavy, light, and SILAC is not always on the medium. It can be like labeling too. So you label one with a heavy label, one with a light, and be able to explain what SILAC does. It's all about seeing a difference in intensity between two proteins, same proteins from different samples. The heavy might be the disease state, or the heavy might be the herbicide resistant state, and the other one's a control. And we want to see, how do these two things change? What is it about the herbicide resistant plant versus the non-herbicide resistant plant that gives it resistance? Doesn't appear to be genetic as far as we can sequence it, and we don't see any major differences, like it didn't pick up a new gene. But the point is, well, maybe it's the expression, which might be a new concept that we talked about, right? That you don't have to necessarily have a difference in genes as far as what's present, but if you have a difference in expression of those genes, that can give you a selective advantage, not merely picking up a new gene or mutating a gene. Yeah?
What does SILAC stand for again?
Oh, it's SILAC. What is it called? That's SIL. I don't remember.
Stable isotope.
Okay, so
labeling
that's-
Stable
amino acid.
isotope label
Amino
affinity
acid.
chromatography.
Yeah.
Is that what it is? SILAC. Stable isotope labeling, and I forget what the AC is. Okay, so that's all you need to know, because that's
Wait, what-
all the things that they are doing is basically labeling them and then using slight differences in mass to figure out their intensity differences. Now, why do they use like radioisotopes, like deuterium versus hydrogen? Because when you label something, you don't want to change their ability to fly differently in the mass spec, right? If you were to say, okay, here's what I'm going to do. I'm going to label this one with a totally different molecule than this one. That could influence their ability to go into the gas phase artificially. So it has to be labeling with something that's nearly identical. So if you use just different isotopes, deuterium versus hydrogen, it doesn't change their ability to go in the gas phase. It's the same molecule, just one slightly heavier than another. Because you have to make sure that you're not changing their ability to go into the gas phase in the same way. Does that make sense? So, for instance, you might devise, okay, here's a protein, okay? Here's the same protein, let's say this is normal disease. Well, let's say I'm labeling it with something to make it heavier. Okay, I'm going to label it with something like that, and I know how much heavier that is. If we were to label with a different molecule, we would say, oh, this has a different mass, I'm labeling it with this different molecule. That molecule is different in its chemical nature, wouldn't you agree, than a long chain fat versus something like this? That can influence its ability to go into the gas phase. So intensities are also related to the ability to go into the gas phase. If you're labeling them with the same molecule, but one has maybe different stable isotopes, like deuterium versus hydrogen, they will fly the same way, because it's the same molecule, just one has now a heavier isotope labeling on it. Does that make sense? Okay. So that's why they are always usually working with very similar labeling agents, and they're just different in whether they are contain deuterium or hydrogen or whatnot. Because you don't want to artificially cause one to appear downregulated because what you're attaching to it actually inhibits its ability to go in the gas phase. So those are considerations when these people are developing these tagging tools. We got to make sure that between the two samples we're testing, that what we label them with doesn't bias one sample going into the gas phase versus another. They need to be able to go to the same extent. Does that make sense? So we have MSMS, 2D isoelectric focusing, and this idea of SILAC. I would kind of focus on this one where we use cysteines to label them, one with a light, one with a heavy. Okay. Because that's the major way that they do these things. Okay. Did I test you on that stuff? I think I made it through all the chapters on those. Okay. All right. Then we talked about protein-protein interactions. What do I want you to know from protein-protein interactions? What a pull-down is. Okay, and I gave you a test question on that. So this is the common way. We have a bait, right? A protein we're interested in knowing what it interacts with. Okay, and then we have the prey proteins. Okay. And the prey proteins are the ones that interact with it, actually. So we don't know what interacts with it, but we're interested in this protein. A pull down is basically having a way to put that bait on a hook, something that allows you to pull it out of solution so that the prey come along with it. Okay? So you'll want to understand this idea of a pull down. Okay, yeast hybrid system is not great because you have to be able to use it in yeast and all these other things. This is probably the more common one, is this pull down. That you have a protein of interest, you have some kind of hook to something, in this case it's avidin, and then now when you have it mixed with things, right, the avidin allows you to pull it out. Okay, you can bind it to streptavidin, or to biotin actually. Biotin will bind to it, you can pull those out, and then whatever has bound to it comes along for the ride. So the prey comes out with it. That's the basic concept there. How do we prevent things like, well, dissociation? Because remember, protein-protein interactions are non-covalent, concentration dependent. Okay? And so how do we do this? We can do things like chemically cross-link them to permanently connect them so that things that were associating with it, you know, when we start to purify, they don't just fall off and fall away. And when Dr. Chowdhury came, he talked about this. So, pull down, how do we prevent things like loss of non-covalent interactions? We can cross-link. Don't need to know the cross-linkers. I would like you to know that approach. We know what interactomes are, right? That's ultimately what we're trying to do with the pull down. What is interacting with what? So we good there? Know how to explain those, draw up pictures, schematics. Okay. Protein-protein interactions, post-translational modification. Okay, we talked about 3 different kinds of post-translational modification. We talked about ubiquitination, we talked about phosphorylation, and we talked about glycosylation, right? Those are the 3 biggies that everybody cares about. Let's see. Did one of your groups do something on
Yes.
ubiquitination?
So,
What was that?
that was our group,
Yeah. What was yours group on?
ALS and mice.
Okay, Alice and mice. So, let's focus on from here. And we talked about the proteasome. They're all basically done in the same way. So what's the take-home message from this chapter? We want to decipher, okay, post-translational modification. We need, one, a way of enriching, right? It's basically up here. We need a process of separating things that have been post-translationally modified so that we get rid of all the background noise and focus on that. So enrichment, usually that's a pull down of some sort. And then afterwards, what do? Basically MSMS, okay? So post-translational modification, how do we start to investigate that? We need first some way of enriching it. Number 2, we just do MSMS. That's why that chapter is so important. That's why it's so important for you to remember. That wasn't really a chapter, that topic. All right, so that's it. Let me see, questions I could ask from these different things. Let's just focus on those because then nobody has an upper hand. I'm not gonna be able to ask a question from each one of these. Now looking back on it. Sound good?
Yep.
All right, but hopefully this class has been useful, and I thought that, and you guys, you know, you've left your reviews. The hope is that the beginning part allowed you to look at the papers that you investigated and the topics, and you understood what you were kind of reading about. You know, in learning, actually most of your learning comes if you're going on to graduate school and things of that nature. Reading papers like you read, okay? And then trying to, based off your background knowledge, decipher and understand what they're talking about. If you notice, a textbook, reading a textbook is very different than reading a paper, right? The technical terms, all this stuff. And so the difficult part in transitioning from a bachelor's degree and then going into research, going into that, is this jump. The way we read things and the way we present things is totally different from a technical paper to a textbook. The textbook is basically summarizing the main points that all the research that's been done has discovered. When you go into the technical papers, what is it? It's giving you nitty-gritty details, there's new terminology, acronyms, all these things. But that's where you really will start to learn. It's really, you gotta teach yourself, you gotta look at the paper, you read something, and it's like, I have no idea what that is. And they don't give you a key. You have to go back to another paper and read it to understand that paper. And then that usually leads you back to say, oh, I don't understand something from this paper. You start to go back in time and read all these papers. And really that's where, I was told when I was a graduate student, an hour in the library will save you 10 in the lab. Because probably somebody's done a lot of this stuff before. Make sure you read all these past papers because somebody will have this little tidbit of information that's going to be helpful. Oh yeah, we tried this and it didn't work, okay? So as you go on, that's what you want to do. Spend and invest time looking at research papers before you move on to really what you want to do. You want to understand what's been done past, so you understand what may be useful in the present as you're working on something. Does anybody have any questions? Is there anything that I need to clarify?
Yeah, right, real quick. You said don't focus on the presentations? Or
Yeah, don't focus on
Okay.
it. They're so specific, I'm not gonna do that. And they use the same techniques. I would rather you focus on those techniques.
Okay.
You know, MSMS, ubiquitination, you know, I think, is that the same figure that I used? Okay. I was like, wait a second, that looks familiar. But hopefully this was a useful class, because I think this going forward is what's really gonna unlock, you know, the future of things that are really important to us. Medicine is important to us. We all wanna live, hopefully we all wanna live longer and see our families live longer and things like that.
Yeah.
And in order to tackle a lot of these problems, you understand that it's not just understanding the biochemistry at this protein level, one protein does this, but it's now understanding the accumulation and expression of different proteins and how that can influence things like health, things like antibiotic resistance now. Hopefully your eyes have been open to this new concept that it's not merely just one gene, one protein, one mutation, one resistance. It's things that are already there. It's the nuanced changes in expressions of existing genes that can really allow something to become tolerant. And then once something's tolerant, then it can become resistant because it had just the right expression levels to make it through something. It didn't die. So what happens after it sees this drug and just makes it through? Upregulates the expression of those genes that protected it in the first place. Then you go through the next round of chemo, the next round of trying to use a herbicide. What happens? It isn't going to work because now it's primed to be protected, which is probably different than what you've learned. It still can be the gene, you know, mutation thing, but I would say the more adaptable approach, the more rapid approach is just these nuanced changes in genes that already exist, which goes into things like epigenetics and stuff like that. So things that aren't in the code but kind of are. Right? It's like, that's really hard to understand. You have the genes there, but they're not expressible, or they are expressible. So hopefully it also shows you that we don't understand everything yet. It's not as simple as maybe the textbooks make it to be. And I don't think they're lying to you. They're starting with the simple so that you understand that level. Then you can start to branch out and start to add more layers to it, okay, and put the puzzle together. Cool. Any questions?
Why do you have London weather on your helmet?
Because that's what brings up when they gave me this computer. So, yeah.
London.
Don't judge me based off of what's on my computer
I
screen.
wasn't judging you, I was just stating a fact.
Because I care about our brethren over in London, okay?
Who cares
I
about
want
Big
to know
Ben?
what they're doing.
About Big Ben.
Yeah, Big Ben. I want to know how Big Ben's doing. The Thames River, I want to know. Is the Thames, is it doing okay?
I've been super dumb for a while, but I only recently found out that Big Ben is not the name of the tower.
Okay.
It's the name of the hotel.
It's the name of the
Tower is called
Oh
What's it
yeah,
called?
man, I was serious. Yep.
The what?
The tower is actually called the Elizabeth Tower. The bell is called
I know.
Big Ben. I think. I want to look that up. So I'm not saying false information.
Yeah, that's
I think that's it though.
I always thought it was just like,
Yeah, I
oh,
know. That's so
It's
good.
just like then you know that the true Londoner from a true.
yes, that's what movies do to you. It's okay.